Embosphere microspheres, the exciting new class of embolic from BioSphere Medical, the company dedicated to innovating embolotherapy. In the next few minutes, you'll learn about this innovative new embolic, which is designed to set new standards for control, precision, and ease of use in the treatment of hyper-vascularized tumors and arteriovenous malformations. Embosphere microspheres not only look different than first-generation particles, such as PVA, they are different. Now let's take a close look at Embosphere microspheres and the key features that will enable them to make a difference in your practice. The first key feature is that Embosphere microspheres are round. Unlike any other embolic, they are the same shape as the vessel lumen, which enables them to provide a complete mechanical occlusion. Due to the chemistry and irregular shape of PVA particles, occlusion results from a combination of particle aggregation and thrombus formation. This can result in an incomplete and non-durable occlusion. Secondly, the round design enables Embosphere microspheres to be precisely calibrated. Spherical particles have a single axial dimension. Their uniform geometric shape allows them to be sized much more precisely than irregularly shaped particles. The accuracy of the stated diameter range of Embosphere microspheres allows confident selection of the size required for targeted, effective embolization. Because of their irregular shape, non-spherical particles, such as PVA, have a wide variety of axial dimensions. This limits the accuracy of calibration. Embosphere microspheres' round shape and a proprietary manufacturing process provide more precise sizing for procedural accuracy and efficiency. The differences in mechanism of embolization between Embosphere microspheres and PVA were demonstrated in a sheep study by Dr. Jean-Pierre Pelage at Hospital Lariboisière in Paris. As you can see, the study showed that it required 1 to 3 Embosphere microspheres to occlude a vessel versus 10 to 20 PVA particles. Further, the study showed that the size of Embosphere microspheres highly correlated to the diameter of the occluded vessels. On the other hand, PVA particles, no matter what size was selected, up to 600 microns, occluded various diameter vessels in the same place. The overall implication of the study is that Embosphere microspheres provide the clinician with the ability to perform more targeted, controlled embolization. The third key feature of Embosphere microspheres is that they are compressible. They have elastic properties that allow compression of up to 33%. Temporary deformation, combined with Embosphere microspheres' round shape and hydrophilic properties, facilitate remarkably smooth passage, even through microcatheters. PVA is not compressible. Especially in small catheters, PVA may aggregate in the catheter, making successful injection difficult or impossible. In addition, when subjected to compressive forces, such as injection through small diameter catheters, PVA may break up into smaller pieces. When injected into the vasculature, these improperly sized PVA particles can cause untargeted embolization. The fourth key feature is that Embosphere microspheres are made from a well-established non-resorbable material with cell adhesion properties that result in a durable occlusion. They are made of a cross-linked trisacral polymer impregnated with gelatin. Trisacral has been used for years as a base material in chromatography. Trisacral impregnated with gelatin has also been used as a microcarrier of cell cultures since the 1980s. The role of gelatin in Embosphere microspheres is to facilitate endothelial adherence. This further ensures a more complete and durable occlusion. Finally, cross-linking the polymer results in a more non-resorbable embolic material. The material's inertness, cell adhesion properties, and biocompatibility have been well established. As you can see, this tissue sample shows that PVA particles have been partially resorbed over time. The vessel is incompletely occluded, with thrombus filling the voids between the irregularly shaped PVA particles. These tissue samples show completely occluded vessels 6 months and 6 years after embolization with Embosphere microspheres. Note that the Embosphere microsphere remains unchanged. Now let's compare and contrast the injection of PVA and Embosphere microspheres in the vasculature. As stated by Andrews et al. In the May 2000 issue of JVIR, experience with embolotherapy indicates that the production of parenchymal ischemia requires stasis of blood flow at the level of the tumor cells. Thus, incomplete embolization of arterial inflow, including failure to embolize significant collateral vessels, would be expected to give a substandard result. PVA injects as a slurry of irregularly shaped particles. Its propensity to aggregate can make smooth delivery difficult to achieve. Further, because of its irregular shape, PVA can aggregate in the vasculature, producing an unwanted proximal occlusion in the main vessel feeding the tumor, and possibly embolization of untargeted tissue. Embosphere microspheres inject very easily due to their uniform shape, compressibility, and hydrophilic surface. The spherical shape and hydrophilic surface of embosphere microspheres allow them to take full advantage of hemodynamic flow in seeking their target. The accuracy of the stated diameter range of embosphere microspheres allows confident selection of the sphere size required for targeted, effective embolization. This provides the clinician with the ability to target the microvasculature of the tumor and then upsize the spheres to shut down the feeder network while maintaining patency of the main feeding vessel. Embosphere microspheres were initially marketed in 1994 and received a CE mark in 1997. They have been used to treat over 7,000 patients worldwide in applications including meningiomas, spinal AVMs, epistaxis, and uterine fibroids. In the United States, Embosphere microspheres have received FDA 510K clearance for the treatment of hyper-vascularized tumors and arteriovenous malformations. Pursuant to a separate investigational device exemption granted by the FDA, BioSphere Medical is conducting clinical trials in the United States for uterine artery embolization. Clinical studies are ongoing. Embosphere microspheres, the new class of embolic from BioSphere Medical.